Okay, good afternoon everybody. So as the slide says, I'm Graeme Moffat. I'm the co-chair of the HESI DART committee. I co-chair that with Wafa Harouk from the FDA. And I also want to give a big shout out to Connie Chen, who's doubling as a photographer at the back there, as well as Oscar Bermudez, who really keep us honest on a day-to-day basis and really are kind of the oil and the fuel that drives this committee forward. We are involved in many programs, and they do a great job in just making sure we're meeting timelines and are staying focused, so I do want to thank them very much. I also want to thank Tim and Cyril for their opening, because this is actually a really nice example, I think, of convening, translating and protecting, or predicting and ultimately protecting human health. And it's quite a nice segue into the next section when we're talking about confidence and data around decision making. So before we get into the detail of this, some of you may or may not be familiar with the neonatal FC receptor, but the genesis of this is really around appreciating that when we think about pharmaceutical products and even environmental molecules, but particularly pharmaceutical products, we're really talking much more than just about traditional small molecule chemicals that we all probably grew up with. As many of you will know now, we deal with multiple modalities, monoclonal antibodies, and many other biological platforms, most of which contain an FC containing moiety to it. And so I guess the basis for this program is really trying to understand the biodistribution, both from an efficacy perspective, but more importantly probably from a safety perspective. Where do these molecules actually go? And Jan Willem van der Laan, who's sitting at the back here, published a very nice review, about 2009 I think it was, looking at really what do we understand across species, both non-clinical species as well as humans. What do we understand about the ability of these molecules to get across the placenta to the embryo and developing fetus? So in addition to what we do know, probably more importantly, what do we not know? So this project was a part of quite a large effort to try and fill some of those data gaps. So what were we actually doing, and why were we doing it? As I've kind of alluded to, is really this is all about trying to get a better understanding of how molecules get across the placenta to the fetus, and really understanding that in both the species we do our non-clinical safety testing in, but also we had access to some human placental tissues, so we could do a direct compare and contrast across those species. And ultimately the reason for that was to enhance our ability to do human risk assessment. So if we can understand better our models that we're using to predict human toxicity, or potential for human toxicity, and actually compare that to some human data, then we should be able to be in a much better spot. So what do we know? So I guess for obvious reasons, there isn't a wealth of data from people who have done very in-depth interventional studies looking at the transfer of, if you think of monoclonal antibodies across the placenta in humans. So a lot of the data we're relying on, and there isn't very much of it, is just based on endogenous immunoglobulin. So as many of you will know, the developing embryo and fetus can't make its own immunoglobulin, so it depends entirely on its mother to provide IgG and a little bit of IgA to confer passive immunity. So we know immunoglobulins do get across the placenta, but when does that actually happen and how much does it occur? So one of the few papers that looked at this was actually from 20 years ago. So early in gestation, there's the exosalomic cavity which surrounds the amniotic cavity to the fetus. You can see that on the top right of this slide here. And they sampled from as early as day 6 in gestation through the remainder of first trimester to really understand how much maternal IgG was there. And they were able to detect some, but the levels were very low compared to what's present in the maternal circulation. And I guess it's data like this that really form the basis of the comment in ICH-M3R2, which says it is understood the transfer of monoclonal antibodies during organogenesis is low. But we don't have any data for humans from a monoclonal antibody perspective early in gestation, in this critical period when we think the embryo is at its most vulnerable. And also, as Jan Willem pointed out in his review, we didn't have a lot of data in animals either. So several of us, actually independent of the HESI group, but we brought some of this data together to focus on this program. We looked at the transfer of immunoglobulin monoclonal antibodies during gestation, and particularly focusing on the early stages. So this is one example of data that we published from Amgen when we looked at non-human primates and rats. And so if we look to the left-hand side of both of these graphs, which is the equivalent stages I was showing you from the human data, so in the primate, gestation day 35, so organogenesis finishes around about day 45 to day 50 in the primate. You can see that we do have transfer of, this is an IgG2 monoclonal antibody. And similarly, down below that in the rat, gestation day 10 and the gestation day 13, we can also detect in the embryonic fetal blood and or tissue. And the proportion is fairly similar to what was reported in humans, i.e. it's about 1% of what we find in the maternal blood, so that which is the blue bar. And similar work's been published by colleagues at Pfizer, Lilly, and some other companies, and all telling a fairly similar story. So we do know that we are getting transfer of monoclonals early in organogenesis, but in the absence of getting more data on the human, similar to this, because obviously there's not a huge appetite for doing this type of study in the human, can we understand a little bit more about the mechanism by which these monoclonals get there, as a way to actually get a window on the similarity and or differences between humans and the non-clinical models. So before we get into the data, just a brief introduction of how FcRN-mediated transfer works. So when you look across species, there are pretty significant structural differences in the way placenta is composed and made up and across. On the left-hand side here is rat and rabbit. On the right-hand side is non-human, primate and human. But although many of the structures, and I'm not going to get in a lot of the detail, for those of you who want to see this, there's a really nice review that John DeCesso and some colleagues from Pfizer wrote, I think about 3 or 4 years ago, which really goes into this in a lot more detail. But if you draw your eyes to the figure kind of at the bottom right there, so although the structures are different, the actual mechanism by which this process works is fairly similar across species. So at the bottom, you've got the immunoglobulin circulating in the maternal blood. It then gets endocytosed into these vacuoles. Then you get release of these lytic enzymes from the lysosomes, which acidifies the environment. And that lower pH, then the immunoglobulin will then bind to the FcRN and then can be pulled across the placental barrier to the fetal circulation. And when it gets across to the fetal circulation at neutral pH, so it's a pH-dependent binding, then the IgG gets released from the FcRN and then is able to get into the fetal blood. So that's the basic process by which it works. And really the focus of this work was to really understand how much FcRN is expressed, and more importantly, how much is expressed at different stages of gestation, and particularly early on in pregnancy. And leveraging the full weight and, I guess, power of HESI, this truly was a very integrated collaboration. So this was the number of companies and different approaches that we used, as well as the species. So you can see that we used all of the main non-clinical species that are used for these types of studies. We had access to placental tissue, we had access to sub-yolk sac in the majority, and as I mentioned, we had access to human placental tissue, including first trimester placental tissue too. And you can see the people that were involved in doing the work. The vast majority of the analysis was done using a quantitative mass spec approach, which is a proteomics-based approach. We also had a couple of other techniques, including Westins and immunohistochemistry, to understand both the quantity as well as the localization of the expression. So if we start with the most important species, which of course is the human, so as I showed you earlier on, consistent with that exposure data, we were able to detect, and I say we here, this was very nice work done at AbbVie by Susan Westmoreland and Belen Tornese with samples provided by McMaster University, doing immunohistochemistry and placental tissue from each of the 3 trimesters, and you can see expression of FcRN all 3 trimesters. It perhaps slightly changes in distribution, so there isn't many in the syncytial trophoblasts, which are the cells at the outer edge of these diagrams, and it increases a little bit in intensity, but broadly speaking, it's fairly similar across each stage of pregnancy, which again would support the fact that we are seeing transfer of the immunoglobulin. And bearing in mind as well, obviously the placenta increases in size very markedly as we go through gestation, so although on a per unit basis the expression may not be higher, obviously the total FcRN expression is going to be much higher towards the end, and therefore more ability to get antibody across. So that's the human situation. If we move to the non-human primate, And this is using two different methodologies here. So on the left-hand side is a quantitative mass spec Beck approach, and this is actually from the same samples as the placental transfer data I showed you earlier on. So we're able to compare and contrast how much antibody was getting across to the actual FcRN expression. And then on the right-hand side is a Western blot using pretty much the same time points. So the left-hand side was data that Amgen created, on the right-hand side was data that Chris Bowman at Pfizer generated. But the take-home message from this slide is that, similar to what we saw in humans, we're seeing expression of FcRN in the placenta at all stages of pregnancy, including during organogenesis, which is towards the left-hand side. And then the other take-home and somewhat surprising result was it doesn't seem to change too much as you go across gestation. It stays fairly flat. But again, bearing in mind the placenta is going to be much bigger, so this is expressed as a per unit of per microgram of tissue on per microgram tissue basis. So that raises one question. That seems a little bit odd because it doesn't quite match what we saw in the exposure data, which clearly does seem to increase as we go across gestation. Perhaps one insight into that is when we look at some of the other species. There's a lot of data in this slide, but let me just talk to the highlights. So we've got 4 species, rat, mouse, guinea pig and rabbit on this slide. And the take-home message from this is that again we're seeing FcRN expression early on in pregnancy. But one of the key things is we're seeing much higher expression in the inverted yolk sac compared to the actual placenta. So if we focus on the top left here, which maybe tells it most clearly, this was data generated by Lilly, Bill Breslin's group at Lilly. You can see the blue bar, which is the yolk sac, and the red bar, which is placenta. Again, the placental expression stays fairly flat, but yolk sac is much higher and definitely increases as you're going across gestation. And you see a fairly similar picture when you look across the other species. The graphs on the right are maybe a little bit confusing because the data is plotted in a different way. They're plotted as a ratio of yolk sac expression to placenta, but the same message holds. We're seeing higher expression in the yolk sac. So, a lot of data, and for those of you who want to hear more, if you're coming to the Teratology Society meeting later this month, we've got a half-day symposium on all of this. So I've tried to cram all of that data into 15 minutes, so hopefully I've whetted your appetite if you're coming to that meeting. So the take-home conclusions from this work was that we've been able to detect FcRN expression from early on in pregnancy, and it seems to match, at least on a qualitative basis, with the data we've got around placental transfer of antibodies themselves. And I guess the most exciting part of these data is we actually have got some human data that we can put alongside our non-clinical species, and it really gives us confidence, at least on a qualitative basis, that we're seeing some similarities between the preclinical species and human. So it gives us a little bit more confidence in terms of actually trying to interpret potential effects we've seen in the non-clinical species and their relevance for humans. On a purely scientific basis, I guess a surprising piece out of this was the higher expression that we saw in the yolk sac compared to the placenta, and some further work we've done to explore that. And then obviously, like everything in science, we haven't answered all the questions. We clearly haven't looked at the function of the FC receptor in any of these studies. But perhaps also, as we think through what we haven't addressed, we've done a little bit of work on this, but not a lot. In addition to FcRN, we know that a lot of other Fc gamma receptors are expressed in these areas. And so it is intriguing to speculate what the potential role of some of these other receptors may be to identify some more subtle differences. So I think this project has made some significant advances in our understanding of this, but as ever with science, it still leaves us with some answers, some questions to be answered. And then just to highlight all of the people involved, I'm not going to read this slide, but you can see here the number of companies that were involved in this, as well as academic institutions. This truly was a very collaborative effort, and I think generated a huge amount of data, which advances our understanding of this whole process. Happy to take questions.
Thank you, Graeme. Questions for Graeme? And Bill?
Should we revise F6? On what basis? I don't think so, no. What are you driving at? Where do you think it's lacking?
Do you want to use the microphone, Jan Bill, please? Actually, this is being recorded, so if questioners could go to one of the microphones, that would be very helpful.
You say that the FC receptor is there from the very beginning, and so transport is likely from the very beginning. That's not what was observed. But so the concentration in the fetus is very low in the first trimester. So for me, it's puzzling.
It's puzzling, and I guess that's why I'm alluding to, obviously these data don't speak to the function of those receptors. We certainly can detect they're expressed, but as are there differences in functional capability. I mean, I think the way we tend to look at this is, obviously we've tried, the first step was trying to understand how much antibody gets across, which we've got some insight to. This was to try and understand some of the mechanism. You go into these kind of experiments thinking it would be very simple, it would just match up one for one. It doesn't quite look like that, so it's certainly more complicated, but I think that the exposure data is very consistent with certainly anything that's in any guidance, and it comes down to a quantitative assessment of what the exposure was in the maternal data. There was a nice review article that Chris Bowman was the first author in back in 2013 or thereabouts, where we looked at across multiple different FC containing biotherapeutics. And one of the interesting things there, you seem to be when you did a correlation of maternal exposure, almost at every stage of gestation, you could make a pretty good prediction of what the embryonic and fetal exposure was going to be as well.
Hi, I'm David Hutto from Charles River. Related question on the immunochemistry that you displayed there. To my pathologist eyes, it looked like in the early stages, the expression was not really in the vessel associated.
Correct.
It was more on stromal cells, some macrophages maybe or something, and then at the third trimester it was very clearly associated with vessels. So yeah, the expression's there, but if it's not part of endothelial expression, it's probably not functionally pulling antibody out.
I think that's a really important point, and not being a pathologist, I didn't want to get too much into the description of the slide, but you're right, the vascular expression is pretty much non-existent at the early stages, and the most robust cell, as you've already pointed out, is the macrophages, the so-called Hofbauer cells, which seem to be the strongest expressing cells in all stages of gestation, but clearly the distribution. And I mean, another paper which just came out in the last couple of months was looking at mini pigs and looking at FcRN expression, and as you know, mini pigs have not been used for developmental tox studies because it's believed that there's no exposure to the fetus, but yet they were able to define quite substantial FcRN expression at different stages of gestation there. So I think it just speaks to the more complexity than just looking for expression level. It's the distribution, it's the functionality of that receptor. Yeah.
Quite different question. This seems to be focused on biologics, biotherapeutics containing FC. But for biologics, we mostly are focused on F and B and the binding site of our biotherapeutics, not the FC. So the presence of the target is far, I think, more critical than just the ability to transfer a biologic across the placenta. I'm not sure what was the driver for this focus on just transfer and expression of FcRN. And is there anything planned to address the other part if this is meant to be, expand the species use for biotherapeutics? You can elaborate a little bit on this. It's a little bit unclear to me.
So, I mean, you're absolutely right. Obviously, we developed these biologics largely focused around the FAB region for target interaction. The focus of this, though, was the mechanism which is understood to be the primary mechanism by which biologics or any molecule containing an FC region is going to get across the placenta. So that's target independent. This is a transfer process, and also explains some biodistribution of the biologics themselves. So in a lot of companies, they will do a lot of the FcRN binding early on to understand, how strong is the binding and what's the ability for it to actually get to where they need it to be, to where the target actually is. I think obviously there are other FC receptors involved in that process, but so target, we're not ignoring target expression, but this was a specific question to understand the mechanism by which these molecules get across the placenta.
There'll be time for one last question.
Okay, so maybe to take that question one step further, it's very
Yeah.
extent can we translate these findings regard to finding potency.
So as I mean, there was the paper just came out last month with an anti-TNF where they don't have an FC region, and they were finding they were not able to get across the placenta at all. So I think, putting these two questions together, this process is clearly more complicated than we first thought, but at the same time, the basis of it seems valid, that it's an FC-mediated process by which that event occurs. Does that answer your question? Yeah. Okay.
Thank you very much.